Tell.
The history and advances in cancer immunotherapy. Hey everyone! Welcome back to our channel. Today, we're diving deep into a fascinating and rapidly evolving field, cancer immunotherapy. We'll explore its history, recent advances, and the exciting potential it holds for treating various cancers. So, grab your favorite snack, sit back, and let's get started. First off, what exactly is cancer immunotherapy? Simply put, it's a type of treatment that uses the body's own immune system to fight cancer. Unlike traditional treatments like chemotherapy, which directly target cancer cells, immunotherapy aims to enhance or restore the immune system's ability to recognize and attack tumors. The journey of cancer immunotherapy began decades ago. In the 1980s, researchers like Dr. Steven Rosenberg pioneered the use of tumor-infiltrating lymphocytes, or TILs, which are immune cells that have entered a tumor. These TILs can be extracted, activated, and then reinfused into patients, showing promising results, especially in melanoma. Fast forward to the early 2000s, and we saw the introduction of engineered T cells, including the development of chimeric antigen receptors, or CORs. These COR T cell therapies have been game changers, particularly for blood cancers like acute lymphoblastic leukemia. So, how does immunotherapy actually work? Well, it can take several forms. One approach is checkpoint inhibitors, which block proteins that prevent T cells from attacking cancer cells. Think of it like taking the brakes off a car. Once those brakes are off, the immune system can speed ahead and target the cancer. Another method is adoptive cell transfer, where T cells are harvested from a patient, modified to better recognize cancer, and then infused back into the body. This personalized approach tailors the treatment to the individual's specific cancer profile. T cells are crucial players in cancer immunotherapy. They come in various subtypes, each with unique functions. For instance, some T cells can directly kill cancer cells, while others help regulate the immune response. Recent studies using single-cell RNA sequencing have revealed the incredible diversity and complexity of T cell populations within tumors. Researchers have discovered that some T cells in the tumor microenvironment, or TME, can become exhausted, meaning they lose their ability to fight cancer effectively. Understanding these dynamics is key to developing more effective therapies. Speaking of the TME, it's a complex ecosystem that includes not just cancer cells, but also immune cells, blood vessels, and various signaling molecules. The TME can influence how well the immune system can respond to cancer. For example, some tumors create an immunosuppressive environment, which helps them evade detection by T cells. Recent advancements in single-cell technologies have allowed scientists to map the TME in detail, revealing how different immune cell types interact with cancer cells. This research is paving the way for more personalized and effective immunotherapies. Despite the progress, there are still challenges to overcome. Not every patient responds to immunotherapy, and researchers are working hard to understand why. Factors like tumor heterogeneity and the presence of immune-suppressive cells can impact treatment outcomes. Looking ahead, the future of cancer immunotherapy is bright. With ongoing research into combination therapies, novel targets, and improved patient stratification, we're getting closer to making immunotherapy a standard treatment option for more cancer types. Now, let's talk about cancer vaccines. These aren't your typical vaccines that prevent diseases. Instead, they're designed to train your immune system to recognize and attack cancer cells. They work by using tumor-specific antigens, which are unique markers found on cancer cells. By introducing these antigens into the body, we can kickstart an immune response, helping the body to identify and destroy tumors. Next, we have oncolytic virus therapies. This sounds complex, but it's pretty cool. Scientists use genetically modified viruses that specifically target and infect cancer cells. Once inside, these viruses replicate, causing the cancer cells to burst and die. Plus, they stimulate a broader immune response, helping your body fight off any remaining cancer cells. Now, let's shine a light on natural killer cells, or NK cells for short. These are a type of immune cell that patrols the body looking for abnormal cells, including cancer cells. They're like the body's special forces, ready to attack and eliminate threats without needing prior exposure to the cancer. They're crucial in the early response to tumors. Moving on to CAR T cells, which stands for chimeric antigen receptor T cells. This innovative therapy involves taking T cells from a patient's blood, modifying them in the lab to express a receptor that specifically targets cancer cells, and then reinfusing them into the patient. It's like giving T cells a superhero upgrade, enabling them to find and destroy cancer more effectively. Next, we have anti-CTLA-4 antibodies and anti-PD-1/PD-L1 antibodies. These are types of immune checkpoint inhibitors. Think of them as the brakes on your immune system. Cancer cells often use these checkpoints to evade detection. By blocking these checkpoints, these antibodies unleash the immune system, allowing it to attack cancer more vigorously. Let's not forget about cytokines. These are signaling proteins that help regulate and direct immune responses. They can enhance the activity of immune cells and improve their ability to fight cancer. Some cytokines are even used therapeutically to boost the immune response against tumors. Finally, we have TCR-engineered T cells. Similar to CAR-T therapy, this approach involves modifying T cells, but instead of using a chimeric receptor, we enhance their natural T cell receptor to recognize specific cancer antigens. This method is particularly useful for targeting tumors that express unique proteins. So, there you have it. From cancer vaccines to CAR-T cells, these innovative therapies are changing the landscape of cancer treatment. They harness the power of our immune system to fight back against cancer in ways we never thought possible. Now, there are several major categories of immunotherapy, each with its unique approach. Let's break them down. First up, we have oncolytic virus therapies. This might sound a bit sci-fi, but it's actually quite simple. Scientists use genetically modified viruses that specifically target and infect cancer cells. Once the virus enters the cancer cell, it replicates and causes the cell to burst, releasing new viruses that can go on to infect other cancer cells. This not only destroys the tumor but also stimulates the immune system to recognize and attack the cancer. One well-known example of this is Talimogene laherparepvec, or TVEC for short, which is a modified herpes simplex virus used to treat melanoma. Next, let's talk about cancer vaccines. Just like traditional vaccines that help prevent diseases, cancer vaccines are designed to trigger an immune response against cancer cells. They work by introducing specific tumor antigens, essentially, markers that are unique to cancer cells, into the body. This helps the immune system recognize and attack those cancer cells. There are two types of cancer vaccines: preventive vaccines, which aim to stop cancer from developing, and therapeutic vaccines, which help treat existing cancer. One prominent example is the Sipuleucel T vaccine for prostate cancer. Moving on to cytokine therapies. Cytokines are proteins that act as messengers in the immune system, helping cells communicate and coordinate their response to threats like cancer. In cytokine therapy, these proteins are administered to boost the immune response. A well-known cytokine used in cancer treatment is interleukin-2, or IL-2, which stimulates the growth of T cells, the immune cells that fight cancer. While cytokine therapies can be effective, they can also cause significant side effects, so they're often used in combination with other treatments. Now, let's dive into adoptive cell transfer, or ACT. This innovative approach involves taking immune cells from a patient, usually T cells, and modifying or expanding them in the lab to enhance their cancer-fighting abilities. These cells are then infused back into the patient. One exciting advancement in ACT is CAR-T cell therapy, where T cells are genetically engineered to express a receptor that targets specific cancer antigens. This has shown remarkable success in treating certain types of blood cancers. Finally, we have immune checkpoint inhibitors. These are drugs that block proteins on immune cells or cancer cells that prevent the immune system from attacking the tumor. Think of them as the brakes on the immune system. By inhibiting these checkpoints, the immune system can unleash its full potential against cancer. Some well-known checkpoint inhibitors include anti-PD-1 and anti-CTLA-4 antibodies, which have been game changers in the treatment of melanoma and lung cancer. So, to recap, the major categories of immunotherapy include oncolytic virus therapies, cancer vaccines, cytokine therapies, adoptive cell transfer, and immune checkpoint inhibitors. Each of these approaches has its unique mechanisms and applications, but they all share a common goal: to empower our immune system to fight cancer more effectively. If you found this video helpful, don't forget to give us a thumbs up and subscribe for more content on cancer research and treatments. And let us know in the comments which aspect of immunotherapy you find most interesting. Thanks for watching, and we'll see you next time.
repel.